Halobacterium salinarum is an example of a halophile. This is an archaea that has several unique biochemicals in and on its structure, on its cells, that we find to be very helpful research tools, as well as unique biochemicals that this organism uses in nature. As the name implies, Halobacterium, well, when it was first discovered, scientists thought it was a bacterium. Halo referring to salt, and then the species name says salt, salinarum. So this is a halophile, prefers to grow in very salty conditions. One of the unique chemicals that's found in Halobacterium is bacteriorhodopsin. Bacteriorhodopsin starts out as a purple color. So when we see Halobacterium growing in aquatic environments, the water looks purple or very bright pink because of the bacteriorhodopsin. The unique thing about this particular pigment that's light sensitive is it allows this organism to adjust its location within the water column based on how much the light intensity is. And what's just the right amount of light. To do so, the bacteriorhodopsin is going to capture those photons of light, and when it does so, it will change from purple to clear to purple again. It's the same kind of sort of flashing or alternating sort of color that this pigment shows that also occurs in the rhodopsin in the mammalian eye. And it's that capturing of photons of light energy and then getting that energy of photons converted to the energy of a movement of electrons and other biochemical structures that allows mammals, including humans, to see. Since the biochemical sort of reaction to photons of light is essentially the same in bacteriorhodopsin, it's a good tool to study how that particular light-sensitive chemical works. And by doing that, then we can see what happens with mammalian rhodopsin. But we can do a lot of preliminary groundwork or preliminary study of the biochemical and electrochemical relationships using this archaea first instead of a lot of mammalian eyeballs. Another unique chemical in Halobacterium is a protein that contains a heme unit that's similar in structure and function to hemoglobin that we find in mammals. It's an oxygen sensing or detecting biochemical. With this oxygen protein or heme protein, Halobacterium can again adjust its position within the water column, not only for light using bacteriorhodopsin, but now with this heme protein also in particular where the appropriate amount of oxygen is present. So this unique organism has these two sort of gifts for us in research, one to study light detection, another to study molecules that can detect and interact with oxygen. Another type of halophile is Haloquadra walsbyi. Haloquadra walsbyi, when first discovered, gave microbiologists a unique glimpse into a unique morphologic shape that's not very common, and that's the square shape. Remember when we went through those examples of just prokaryotic characteristics, we described that usually when we look at bacteria or even archaea, they usually come as spheres or rods or perhaps a curved shaped cell. But that rarely you find other geometric shapes, and this is one of those. That's where you get the name quadra. It's shaped usually in little squares that assemble in groups of 4, and halo referring to salt, so this is definitely a salt-loving organism. And the species name is in honor of the first person to discover these square, salt-loving archaea in the Red Sea in the Middle East, and that was Tony Walsbyi in the 1980s. The image that you see at the top right and in the center top are images taken with dark field microscopes. So you can also see that the images give us a nice appearance or illustration of what these cells, individual cells, look like. And the bottom lower image is again another kind of sort of artist rendition of the individual cells as we might visualize them with a regular microscope. So the artist has kind of colored the background, but these cells also have photosynthetic detecting pigments, or I should say light detecting pigments, that are various colors of pinkish appearance. So the top images are using a specialized kind of microscope, so the cells really don't appear green in nature. In fact, they have more of that pinkish color that we see with some of the salt-loving types of organisms from other parts of the world. They were discovered in the salt pools called Sabkha and can still be found today. Another extremophilic bacterium that's really unique is Deinococcus radiodurans. One of the types of microorganisms that we very likely might find on other planets or moons in our solar system could very well be this type of microorganism. It is a bacterium. Most extremophiles are archaea, but there are bacteria that can be extremophilic, and Deinococcus is an excellent example. For humans, if you're exposed to 5 grays, which is a unit of measurement of energy intensity, essentially, of ionizing radiation in the form of gamma radiation, that can result in roughly a 50% mortality rate. 5 grays can kill 5 out of 10 people if you're exposed. Humans that are exposed to 7.5 grays or higher, it's going to kill everybody. So that gives you an idea of essentially numerically what it would take in terms of ionizing radiation in the form of gamma rays to kill a person. For Deinococcus radiodurans, we have 50,000 grays as our extreme high level of gamma radiation. Anywhere from 5,000 to 50,000 is well tolerated. In fact, Deinococcus was first discovered in radioactive waste materials in different types of mining industries that essentially mine or search for minerals like uranium, or the waste material left over from using them in nuclear power plants. And so this organism thrives in extreme ionizing radiation environments, and as such has been nicknamed by several other microbiologists as Conan the bacterium. You can see it has this very sort of spherical, globular shaped group of cells and a very hardy little organism. Some other terms that we use to describe growth characteristics. Troph means we have an affinity for some kind of nutrient requirement. And then we add a prefix that tells us what kind of nutrient or what type of unique growth requirement. An oligotroph doesn't need a whole lot to eat. Remember, oligo means scant. And so just scavenging a few nutrients, these are organisms that eke out a living in places in the world that nothing else really could grow. An autotroph grows using very simple inorganic compounds like carbon dioxide directly, or nitrates or sulfates. Very simple sources of food. Again, eking out a living where hardly anything else could grow. Heterotrophs are us. Heterotrophs require very complex organic sources of food, and that would include us. There are many types of bacteria that grow in and on the human body that are also heterotrophs. They require fairly complex foods compared to some of their other sort of cousins in the biomicroorganism world. For chemotrophs grow using simply straight chemicals. And so, again, they're often just requiring very unusual kinds or sources of what we would think of as food. Phototrophs must have sunlight for growth. Cyanobacteria are examples of phototrophs. We have different types of halophilic bacteria that are phototrophs. So using sunlight for growth, those are going to be the photosynthetic or the phototrophic microorganisms that we find on Earth. A lithotroph is a rock eater, and those types of organisms, we didn't really expect to find them. If you dig down in the soil or dig down into the rocks of the crust of the Earth, scientists didn't expect to find microorganisms growing there. And of course they grow extremely slowly, but yes indeed, they're converting slowly but surely some of those inorganic compounds found in the minerals of the rocks of the crust of the Earth. A sulfotroph is an organism that uses sulfur compounds for growth, and a ferrotroph grows and uses iron compounds. So microbiologists and scientists in general can often create these descriptive terms that tells us about the growth conditions and requirements for a wide variety of organisms. Barophiles, piezophils. Those are organisms that have either a requirement for high pressure or can tolerate high pressure. Barotolerant bacteria are able to survive, but when you bring them up to the surface of the atmospheric pressures found at sea level, they're not going to fall apart. The barophiles are required to grow at high barometric, high hydrostatic pressures at the bottom of the ocean. Challenger Deep was essentially an underwater spaceship that went as far down as possible in the Marianas Trench, which is one of the deepest places on the Earth's surface in the ocean. The pressures in the Marianas Trench range from 40 to 100 millipascals. Normal atmospheric pressure on the surface of the Earth is about 0.1 millipascals. If we take a look at where Geobacillus can grow, it can easily grow in those pressures found in the Marianas Trench. And in fact, that's often where these types of bacteria and barophiles have been found. So Geobacillus is our classic example of a piezophile or barophile. It means the same thing. Requires really high temperatures, and if we bring them to the surface of the Earth for study, we have to keep them in containers that have that extreme pressure on those cells so they essentially don't fall apart.